Thank you for joining us for today's webinar, Generating High-Quality Genome Drafts from Uncultured Microbiome Samples with a Single Shotgun Experiment. My name is Karina Dill. I work at 10x Genomics, and I'll be your host today. Next slide, please. Before we get started with the presentation, there are a few disclaimers and housekeeping items I need to go over. First, I'd like to point out that all 10x products, including those discussed today, are for research use only and not for use in diagnostic procedures. Today's webinar will begin with a presentation followed by an open Q&A session. The webinar is being recorded and will be made available for on-demand viewing on the 10x website. All attendees will be on mute throughout the session. In order to ask a question, click on the Q&A option at the bottom right of your screen as shown here on the slide. A pop-up window will open where you can enter your question. You are welcome to enter questions at any point during the presentation or Q&A period. Next slide, please. And now it is my pleasure to introduce our guest speaker, Dr. Ami Bhatt, Assistant Professor of Medicine and Genetics at Stanford University. Dr. Bhatt received her MD and PhD from UC San Francisco. She has been the recipient of numerous awards and honors, including the Chin Award of Excellence from the Human Genome Organization and a Damon Runyon Clinical Investigator Award. She is also the co-founder and president of Global Oncology, a nonprofit organization whose mission is to bring the best cancer care to underserved patients around the world. Dr. Bhatt's research program at Stanford is dedicated to characterizing and dissecting the microbe-human interface. Her group applies modern genetic, molecular, and computational techniques to better understand the host-microbe interaction and how perturbing these interactions may cause human disease phenotypes. Dr. Bhatt, welcome, and thank you so much for sharing this presentation today.
All right, thank you very much, and thank you to all of you who are joining me today. Today I'm going to be presenting work that was carried out collaboratively in my lab, the lab of Serafim Betsoglu, and the lab of Ann Dekis. The work was primarily led by Alex Bishara, a former postdoctoral fellow in the lab, and Eli Moss, a graduate student in the lab. So just to start, these are my disclosures. And let's get started. So today I'm going to talk with you about a variety of things that relate to the microbiome and microbiome genomics. We'll start by discussing the trillions of neighbors that each of us lives with. We'll try to ask the question of who's there in the microbiome. I'll try to convince you that there are perils associated with the simplified gut microbiome. We'll go through two mystery cases, a problem and a solution. And I'll end with a vignette that hopefully will convince you that microbes may be evolving in much faster timescales than we previously thought. And of course, we'll close with a section where we think a little bit about what the implications of these findings are. So as you all know, we live in complex symbiosis with trillions of microbes. And while for the vast majority of the work that has been done to date in the realm of translational medicine, most of us have focused on the host side of the equation, it's clear that microbes are important as well. And when one of these changes, for example, the host changes, so does the microbiome. But perhaps when the microbiome changes, this impacts host biology as well. Of course, the microbiome is comprised of viruses, bacteria, and fungi. And the vast majority of these organisms, viruses and bacteria, are haploid. This presents some interesting issues related to the application of genomic methods. When we ask the question of who's there in a microbiome, the majority of methods applied to date to figure out who's there really rely on sequencing-based approaches. Of course, prior to the advent of approaches such as 16S ribosomal RNA sequencing, visual approaches such as microscopy prevailed in the realm of trying to enumerate the microbes that were present within a mixed community. But in the sequencing era, obviously we have a lot of sequencing techniques that we can choose from when we're trying to figure out who's there in the microbiome. The workhorse of microbiome research is 16S sequencing. Of course, 16S ribosomal RNA sequencing is a method that is very high throughput, relatively cost efficient, and allows gross taxonomic classification. And in rare cases allows us to actually classify organisms to the point of genus, and even species. Of course, some of the limitations of this method, which kind of gives us a piece of the puzzle of the genome of an organism, is that it only applies to bacteria and archaea, so we can't classify, obviously, fungi, which don't have a 16S ribosomal RNA, or viruses, which have no ribosomal RNAs whatsoever. But this does allow us to do a lot of gross taxonomic classification, and as many of you know, the vast majority of manuscripts that have been published on the topic of the microbiome to date have used 16S sequencing. With the advent of shotgun sequencing and shotgun sequencing being more affordable, many have walked down the path of doing high throughput shotgun metagenomic sequencing of microbiome samples. Of course, metagenomic sequencing in its current iteration for the most part generates short reads, which requires us to have methods to be able to analyze the short read data that results. In so doing, we can get higher resolution taxonomic classification. Sometimes we can get designations that go down to the species or strain level. This, of course, allows us to look at non-bacteria and non-archaea. So this allows us to look at viruses and fungi. And of course, when we get shotgun sequencing data, we get data for the entire genome or subsets of entire genomes that are present within a sample. And that allows us to do some functional classification of the genes that we're able to identify within a sample. Of course, this gives us a more complete picture of the puzzle, but it's by far complete. One of the things that has been interesting for me to learn is that taxonomic inference from shotgun sequencing of samples is actually quite limited. In my realm, we study predominantly the gut microbiome of humans. When you take a stool sample and you try to do, for example, k-mer-based taxonomic classification using a tool such as Kraken, and as comprehensive databases as we can generate, on average, we can classify about 30 to 60% of the reads that emerge from a shotgun sequencing experiment. Of course, if I talk to my colleagues who study other types of microbiomes, such as the soil microbiome or ocean microbiomes, a much smaller percentage of data can actually be classified, right around, let's say, 3%, such that even taxonomic classification at the genus level is very difficult in some of these samples. So when we think about microbiome work kind of writ large, and we expand beyond just well-studied microbiomes such as the Western human gut microbiome, and we realize that much of the work that we do is essentially reference-free. So we don't have good references for most of the organisms that exist in this world. And indeed, methods to isolate and culture these organisms and iteratively improve the reference database are ongoing and have been productive. But this really can't quite keep pace with the number of organisms that we expect exist in the world. So shifting gears a little bit, I thought I would introduce the patient population that we predominantly study in the lab, and we'll talk a little bit about the perils of a simplified microbiome. So our laboratory is predominantly focused on the role of the microbiome in the setting of non-communicable diseases, such as cancer and cardiometabolic disorders. And for the most part, we study patients with cancer. The main patient population that we study in the lab are patients with hematologic malignancies, such as leukemia and lymphoma. And a final common pathway of treatment for these patients is often allogeneic hematopoietic stem cell transplantation, also previously known as BMT, or bone marrow transplantation. I'll use the terms interchangeably during this presentation. For those of you who are not familiar with what a BMT looks like or how it works, this is basically how it goes. We have patients who are described as the recipients of the transplant who have a very bad hematologic disorder. Usually these disorders can't be cured with chemotherapy alone, and so what we do in preparing them for a stem cell transplantation is we give them a very high dose of anti-cancer drugs or chemotherapies. Often we'll also treat them with radiation. Now, while this has the beneficial effect of knocking down the abundance of cancer cells within their body by several log-fold, it also has the untoward effect of actually destroying every rapidly dividing cellular compartment in their bodies. So this includes hair follicles, so people's hair falls out, but also the hematopoietic compartment, so the compartment that makes red blood cells, white blood cells, and platelets. Now, of course, not having a hematopoietic compartment, or red blood cells, white blood cells, and platelets, is not consistent with having life. And so what we do is we collect hematopoietic stem cells from a healthy donor. Ideally, we try to match that donor. And classically, we would collect those healthy hematopoietic stem cells from the bone marrow, and the bone marrow of the hip in particular. So that's why we used to call this a bone marrow transplantation. We then take those stem cells and we inject them into the recipient, and we wait. We sit around and wait for weeks and sometimes a whole month to have those hematopoietic stem cells engraft and then differentiate into red blood cells, white blood cells, and platelets. And of course, during this time period, patients are subject to a lot of complications because not having an immune system and not having a functional clotting system can be very problematic. So when we take a look at what most of our hematopoietic stem cell patients die of, they don't die of cancer, surprisingly. So these are data from the CIBMTR, which is the International Blood and Marrow Transplant Registry. And what you can see here is only 37% of patients who die after a transplantation, this is in the first year after transplantation, actually die of primary disease or relapse, which means that 63% of our subjects die of treatment-related complications. Now, you may be asking yourself, well, why are we talking about transplant in the setting of the microbiome? It turns out that many of these non-cancer transplant outcomes are associated with changes in the microbiome. So here in this pie chart, everywhere where there's an asterisk, these are complications, infection, graft versus host disease, and other that have been associated with alterations in the microbiome. Indeed, work done by Eric Paymer's group at Memorial Sloan Kettering, led by Yang Tower, showed that alterations in the gut microbiome are actually one of the biggest predictors or best predictors of overall survival in this patient population. In this particular study, Yang and colleagues collected stool samples from patients undergoing transplantation at the time of engraftment. They then did 16S sequencing and characterized the diversity in the samples and found that those individuals with low diversity, here pictured in the red line, had poor overall survival compared to those who had high diversity, which is pictured in the red line, in the blue line. And this, of course, is statistically significant. So what we see, and what Yang and others have also seen, is that post-transplant loss of diversity is common. People go from having a microbiome that looks kind of like the image on the left to having a microbiome that looks more like the image on the right. Indeed, in our own collection, we have 500 patients, sequential patients at Stanford who have undergone stem cell transplantation. We've collected over 1,200 samples, and we've performed low-pass shotgun sequencing. What we find is that over 40% of our subjects end up being oligodominated or having a very low microbiome diversity. So with that background in mind, let's talk a little bit about two mystery cases. The first mystery case is a patient who we saw at Stanford who basically underwent a transplantation. So this individual was a 46-year-old man who had myelodysplastic syndrome, which is a pre-leukemic syndrome. He had progressive disease, so he was slowly developing leukemia, and therefore underwent a hematopoietic stem cell transplantation. In this particular example, what you can see here is a stacked bar histogram of what happened to this individual's microbiome over the course of approximately 60 days. So you can see that prior to the hematopoietic stem cell transplantation, the patient had a relatively diverse gut microbiome, but over the course of the transplantation, the patient actually developed a very simplified microbiome. And while this is just an anecdotal example, this is actually quite common. But underlying all of this, we had kind of a basic question, which was, why did this bug that is here in this light blue in the histogram dominate? Is it a superbug? Is it something like a multi-drug resistant E. coli? Is it evolving very rapidly through, for example, the acquisition of SNPs? Or does it have, more antibiotic resistance genes? We know that these individuals get exposed to a lot of drugs, including chemotherapies and antibiotics. And when we looked at the shotgun data to try and answer this question, we really didn't get a lot of answers. It didn't appear to be one of those common superbugs like E. coli. It didn't seem to be acquiring a lot of SNPs over time. And as far as we could tell, it didn't have more antibiotic resistance genes than your average gut microbe. So we can ask this question another way and just ask, well, what is this blue bug? Obviously, when we try to classify these organisms, we try to compare them to existing reference databases. And of course, we have databases like RefSeq, which now have on the order of 50 or 60,000 different organisms represented. But still, we're just scratching the surface with reference databases like RefSeq, which reminds us that microbiology to date is largely reference-free. And so while we can try to get into the ballpark of what type of organism this might be, in reality, we don't actually have a very closely related organism in the reference database. So while I'll ask you to keep this in mind, this challenge of trying to figure out who's there when you don't have a complete reference database, and I'll introduce another mystery case. And this is a mystery case that was brought to us by Ann Dekas, a collaborator in Earth and System Science. So Ann and her colleagues are ocean microbiome experts. They actually go out into the ocean and they collect deep marine sediment. So this is actually one of the research vessels that they take out off the coast of the Pacific. And they drop these big multicorers into the sea. They then let these multicorers fall to the bottom of the ocean, and obviously they explore lots of different depths of the ocean. And then as you can see in this picture, they basically just take these cores of sediment. So they core into the sediment and they collect samples. At this point, their research is pretty different from ours, but at the next step, it basically converges almost entirely. So what Anna and her colleagues are interested in doing is figuring out which organisms are present in these deep marine sediments and figuring out what they're doing. And they do this predominantly for the purpose of trying to understand carbon cycling and metabolism as it relates to things like ocean changes and climate change. They take these samples and extract DNA. So here, this is probably looking familiar to almost every one of us on the call. And then they try to figure out who's there and what they're doing. Unfortunately, with all of our sequencing technology, it's really difficult for colleagues like Anne and Alma in her group to figure out who is doing what. They can take 16S sequencing-based approaches, and through the process of generating 16S data, lots of taxa have been given names. So we have names for a lot of these organisms. But for the most part, most of these organisms have never been isolated and cultured, and therefore we don't know what these organisms are doing. In orthogonal experiments, Anne and her colleagues can do geochemistry type of experiments. They can do shotgun sequencing and try to functionally characterize the sequences that are present. But they often can't link those ideas of who is doing what. Put another way, they can ask who is there by doing 16S sequencing. They can ask what these organisms or communities might do using shotgun sequencing. But it actually turns out to be very difficult to link who to what. Because in these sorts of analyses, rarely do you get a 16S sequence that actually assembles along with a shotgun data set. So while these scientific and biological questions may be very different, right? We're trying to ask a question of why do certain organisms dominate and how do organisms change over the course of cancer therapy, and our colleagues are trying to figure out which organisms in the ocean sediment do what. They actually have a shared core scientific challenge. And the challenge is, what do we do when we don't have good references? So what we get from our sequencing approaches right now are either, a piece of the puzzle in 16S sequencing, or a sense of, the function of a community using shotgun sequencing. But what we really want is the whole picture. And I would argue that one of the ways that we can get the whole picture is by having completed genomes of these organisms. The state of the art, and a lot of this work has actually been driven forward by colleagues in the environmental microbiome space, is taking shotgun data, so short read shotgun data, and trying to do de novo assembly. And when we do de novo assembly, we can take short reads, as pictured here, and generate more contiguous contigs or longer pieces of DNA. The objective is to generate these metagenome assembled genomes, or these kind of proposed genomes. In reality, what these types of experiments look like is we have a big pile of short reads, we take a computational assembly algorithm, and there are lots of them out there, to generate contigs. And the most popular assembly algorithms for short reads are graph theory-based assembly algorithms like SPADES, MetaSPADES, MegaHit, IDB, AUD, and many others. At the core, while this is a bit of an oversimplification, what these algorithms do is find areas of overlap in k-mers and then essentially daisy chain these areas of overlap together to generate longer contigs. The problem is while we can generate a big pile of contigs from a bigger pile of short reads, we still don't know which contigs belong to which organisms. And so we have this problem of trying to generate these metagenome assembled genomes. The way this is classically done is by taking sequence intrinsic characteristics of these contigs. So you can take each of these contigs. Here, I'll represent them as circles, even though they're not circular. The size of the circle roughly correlates with the size of the contig. So the bigger contigs are bigger circles. Here, I've also color-coded them based on the genus or the family of the top blast hit. So when you compare them to the reference database, what is the closest taxon that you can match them to? You can then organize these different contigs based on features such as sequence coverage or %GC content. And oftentimes what you end up with is kind of a clustering of these different contigs together. So propose metagenome assembled genomes. So the blue things are all probably part of one genome, and the black rings are probably all part of another genome. So this is kind of, from a thinking perspective, a very clear way to go about things. This is actually a real-life example of generating MAGs, and these are samples, shotgun sequencing samples from 2 individuals who underwent stem cell transplantation. And what you can see on the left is an individual who had a complication of stem cell transplantation called graft versus host disease, which is characterized by really bad diarrhea. And on the right, you can see an individual who had an uncomplicated stem cell transplantation and very good GI health after the transplant. What you can see here is, without knowing anything actually about the taxonomy of these samples, you don't even need a reference database in this particular case, you can easily tell that the sample on the right is much more diverse than the sample on the left. So already you can see that by using these types of assembly and binning approaches, you can get a sense of a community structure without ever having to use a reference database. So while this is very advantageous and actually has led to a lot of interesting insights, we do have a problem. And the problem is that while we can see these kind of clouds of contigs that go together, here on the right, I've outlined one in a pink circle, we can't actually prove or generate orthogonal evidence to show that these contigs actually belong together in a metagenome assembled genome. You can imagine that ways to approach this would include single cell sequencing, which might help us identify, for example, RNA transcripts from different contigs that all belong in a single organism. But another way to do this, obviously, would be with longer reads or linked reads. But before we jump into that, I want to pose a question, which is, why are these metagenome assemblies so fragmented? The easy first answer when people started in this field was, well, maybe we don't have enough coverage. And people generated more and more sequencing data to try to improve coverage, to improve this problem. But what you find is that essentially, no matter how much sequencing data you generate, oftentimes these are fixed problems, meaning you cannot overcome them with greater sequencing coverage alone. And really, what it ends up being is an issue related to a structural feature of genomes. And that structural feature is that contigs that are present in more than one place essentially break assemblies. So let's talk about a toy example of this. Here, I've created a toy example of 2 different contigs, or 2 different genome regions. In one genome region, you have a region A, followed by a repeated region that we've called R, followed by another region D. And the other contig, you have the region B, followed by, once again, the repeat R, followed by C. Now, this repeated region is actually shared between these 2 different genomic locations or genomes. And so when you do short read assembly alone, a short read assembler will essentially not be able to differentiate where R actually is supposed to be. It gets essentially confused. And so what typically is spit out of a short read assembler is these 5 contigs separate from one another. Now, if you track back to the actual graph that is generated by these graph-based assemblers, you get a graph that looks like this. So the graph actually knows, or the assembler is able to infer that the repeated region R has either A or B on the left side and either C or D on the right side. The problem is it can't trace a path through this graph properly. Now, repeated sequences abound in nature. An example of a repeated sequence is the 16S ribosomal RNA operon. So for those of you who have done metagenomic assemblies or even isolate assemblies, you've probably ran into the issue where the 16S ribosomal RNA operon usually assembles separately and on its own. Of course, gene duplication also exists, and you have elements such as transposons or insertion sequences, which are jumping genes that are present as duplicated genes throughout individual genomes, but also can be shared between different genomes. So repeated sequences confound current sequencing and assembly approaches. In this particular toy example, which actually we took forward to an actual example in the work that we did to develop linked-read sequencing for metagenomic assembly applications, here we have 5 different bacterial genomes. In each bacterial genome, you have a copy of the 16S sequence, which is in the pink box. And you can see you have variable copy numbers, and the 16S ribosomal RNA can vary in copy number from 1 to over 10. When you combine these together into a mixture and then you sequence, what you end up with is a graph that looks like this. So when you do shotgun sequencing in a metagenomic assembly approach, the graph looks like a big repeated sequence in the middle, which is the 16S ribosomal RNA. Oftentimes, assemblers will collapse small polymorphisms in the ribosomal RNA, and you can see lots of things on the left and lots of things on the right. You'll recognize that this looks very similar, although a little bit more complicated than our toy example. So in theory, long-read sequencing could really solve this problem, and that seemed like a great approach. Unfortunately, one of the things that stymied us in our original efforts to try and do long-read sequencing was that we couldn't get enough high-quality, high molecular weight DNA, or long fragments of DNA from stool samples in order to do this. And this is, of course, in part because most of the methods that we and others have to extract DNA from mixed microbial samples rely on very harsh physical methods, such as bead beating, to extract DNA. So that, unfortunately, while we tried it, didn't work too well in our hands when we were attempting this. I'll just remind you that measuring these repeated sequences, whether they're repeat sequences like the 16S ribosomal RNA or moving sequences like the transposons, is hard. And so measuring these repeated sequences, many of which are involved in gene transfer, thus is difficult. So with that challenge in mind and that goal of trying to improve assemblies in mind, this is when we came to a potential solution. And that solution was the linked-read platform. And so for those of you who are unfamiliar with the linked-read platform, in essence, what it does is it takes high molecular weight DNA or long pieces of DNA and it packs them into gel beads, millions of gel beads. And you can essentially generate barcoded libraries from each individual gel bead. For our application, which was in the metagenomic space, what we did was take a target metagenome, extract DNA using conventional methods. While most of our DNA ends up being quite fragmented, we're able to extract and purify enough high molecular weight DNA in order to prepare a library using the linked-read platform. So we extract DNA and then size select. We then have long DNA fragments. And then we take these long DNA fragments and we use the Chromium platform to generate these read cloud libraries or linked-read libraries. In essence, in each and every gel bead, what we have is a few long pieces of DNA, and each of those pieces of DNA is prepared into a library with a unique molecular identifier or a barcode. And this allows us essentially to backtrack to each gel bead using the barcode information to confirm that sequences generated from that bead were actually generated from the same piece of DNA. So while this was a commercially available technology when we embarked upon this project, unfortunately, we didn't have software for the metagenomic assembly application. Now, the type of data that the 10x approach, the linked-read approach generates looks kind of like this. It's essentially sparse read coverage across a long region. Now, this is very nice, but unfortunately, you don't get deep enough coverage in order to do a sub-assembly from each and every barcode, which would be, of course, ideal in this case. And so what we needed to do was to come up with alternative computational ways to approach this issue and to overcome the challenge of the sparse coverage. And this ended up being a piece of software that Alex Bishara in the lab wrote. The software is called Athena. It's publicly available. I'm guessing that many of you guys have actually had the chance to access it. And what Athena does in essence is it takes the initial scaffold graph that is generated by a traditional short read assembler. We then map all of our reads with barcode information back to the graph. We then take the graph and pool different barcoded reads together that will help us solve the challenges or the ambiguous regions of the graph. So for example, at the region between the repeated region, pictured here in orange, and the areas to the right or to the left, we then do subassemblies and generate subassembled contigs. And in the last step, we use existing either overlap layout consensus assemblers or long read De Bruijn graph assemblers such as Fly to assemble these subassembled contigs into these long contigs. And for those of you who are interested in more details about Athena, you're welcome to contact us or look into the supplementary materials of the manuscript that was published on this topic at the end of last year. So we tested Athena and this approach on first a mock mixture. This was the ATCC20 mock mixture. And once again, remember the challenge in these assemblies is that the 16S ribosomal RNA is a repeated region that often can't be resolved. And to make a long story short, we were able to generate near complete genomes of almost all of the different bacteria in this staggered mixture from a single experiment and an Athena assembly. We then applied this method to healthy human gut microbiomes. And what you can see is we compared it to the synthetic long read approach, which was previously called Moleculone, was provided by Illumina, as well as a traditional short read approach. What you can see in this histogram is that we have both the number of complete genomes and the number of high quality genomes, which is a complete genome plus some additional features such as tRNAs. And what you can see is that the read cloud approach outperforms SLR and short read assemblies in both cases. This is an example of a near complete genome that we were able to generate using this approach. This is the genome presented as a Circos plot of a Phascolactobacterium species. You can see that the genome generated by the read cloud approach actually just has 2 contigs. You can see that the genomes generated by the synthetic long read and the short read approach are much less contiguous, and in the case of the SLR approach, much less complete. In the case of the ocean sediment samples that I mentioned, as you might know, ocean microbiomes are much more complicated and complex than human gut microbiomes. And what you can see here is that the read cloud assembly approach compared to the short read assembly approach far outperformed short read assembly in terms of the number of intermediate quality genomes that it was able to generate that included the 16S sequence. So suddenly, what we were able to do is map in many of these cases, in this case 24 different genomes, we were able to map a 16S marker, which allowed us to infer taxonomy to the remainder or much of the remainder of the genome, which allowed us for the first time to actually start to link who is there to what these organisms are doing. And this has allowed Anne and Elma and their group to start to deconvolute the metabolisms of taxa that have previously been described. So this work was published in Nature Biotechnology at the end of last year, and you're welcome to take a look at the manuscript in the supplement, as well as the Athena software that is deposited on GitHub. But I wanted to come back to the mystery case that I originally posed to you, and that was the case of this individual, this 46-year-old man, I'll remind you, who had MDS, who underwent stem cell transplantation and became oligo-dominated. And as you might remember, one of the questions that we had was, why does this blue organism end up winning? And we think the answer is actually because this microbe was able to evolve. It was able to evolve under interesting pressures. So what we found was that oligodomination by this blue organism was associated in this case with a shape-shifting genome and gene transfer. And I'll take you through the results piece by piece. So When we applied the linked-read sequencing approach in the Athena assembly to these 4 different time points of microbiome samples from this individual, what we were able to do was to assemble a near complete genome of that organism that ended up dominating. That organism ended up being a B. cacchi, a Bacteroides cacchi species, or a new strain of Bacteroides cacchi. And what you see pictured here is a piece of that assembly. So in the blue line that goes across the middle of the slide, what you see is a region of the genome of B. cacchi that was assembled fully using the Athena approach. You can compare that and contrast that to the short read assembly, which is the fragmentary assembly that is present just below the Athena assembly. Above the assembly, what you can see is the gene annotations of all of the open reading frames that are present in that particular part of the genome. What you can see is that there are a variety of different genes that are present. Some of these genes may be familiar to you, some not. But let us zoom into this region a little bit more closely. One of the interesting things that we found was that this center section, which is about 60 kilobases, was assembled into the genome of the organism at time point D, but was absent at time point C, as you can see by this DNA sequence and coverage. What this suggests is that this 60 kilobase segment of the genome actually popped in to the Bacteroides cacchi genome between time point C and time point D, or that there were both isoforms present at time point C, but the version of the organism or the strain of the organism with that region of the genome actually was selected for at time point D. So that's kind of interesting. We actually see, using this Athena assembly approach for the first time, an example of potential active lateral gene transfer. Now, this is a very dramatic example of a big cargo or a big payload of genes being transferred from one organism to another, but there are actually smaller examples of this as well. And then this last example, what I'll pose to you is that it's not how many genes you have, but it's how you use them. So another example of a moving repeated sequence that we found in these data was actually a transposon that seemed to be moving around the genome. Now, transposons or insertion sequences are kind of minimal repeated sequences. These are, of course, jumping genes. They can move around. Typically, they're about 1 to 2 kilobases in length, and they usually encode just a transposase. So they encode a gene that, when transcribed and translated, can mediate DNA copying and pasting or cutting and pasting around the genome. What we found was that in various parts of the genome, when we had the better assembled genome using the Athena approach, that there were regions where the insertion sequence seemed to be absent in earlier time points. So for example, in time point A and B as represented here, but seemed to be present in later time points, such as time point C and D as presented here. You can see that that was confirmed by generating PCR primers to the regions that flank that variable insertion sequence location. You can see that when the insertion is absent, you get a low molecular weight band, and when the insertion sequence is present, you get a high molecular weight band. So this is interesting, but does it really have any functional consequence? This is an example of a genomic region where an insertion sequence was variably present. So in the middle of the region located on the top of the graph here, you can see a gene, which is the insertion sequence. Now, the insertion sequence has an arrow that is pointed outward. That is actually a promoter that can drive transcription of nearby genes. So one feature of these selfish elements is not only can they drive transcription of their own gene, the transposase, but they can also actually regulate genes that are nearby. So in this particular example, what I have here are time points B, C, and D. And you can see at the top time point B, the insertion sequence was absent. At time point C and D, which are below, the insertion sequence was present. So you would expect that when the insertion sequence was present, transcription of the neighboring gene, in this case, the gene outlined in the green triangle, NORM, would be upregulated. Now, NORM happens to be a multi-drug efflux pump. And so you could imagine that upregulating that gene might protect the bacteria from drugs that act intracellularly. Indeed, when we did RNA-seq on these microbiome samples, what you see is that NORM is clearly upregulated when the insertion sequence pops into that location just upstream of the gene. We then went on to isolate and culture various of these strains, and what we find is that, as predicted, when that insertion sequence is present upstream of NORM, the strains that have that insertion sequence present are actually more resistant to antibiotics, in particular intracellularly acting antibiotics such as ciprofloxacin. So this is just an example of what improved assemblies of genomes from microbiomes can allow you to interrogate in terms of functional adaptation in bacteria over time. So I would argue that we now have improved sequencing approaches for metagenomics, and I would argue that long-read and read-cloud metagenomic sequencing can allow us to do de novo species and strain-level classification that can allow us to interrogate non-bacterial data and pathways in more detail. So what? What have we really learned by all of this? I would argue that, of course, human genomics is incomplete without consideration of microbes. They make up a vast majority of our genetic and gene-level variation. We know that a simplified microbiome in the case of the gut microbiome is often associated with disease. We believe that strains matter and that variations in genomes that are slight, that may be SNPs or structural variations, are important to measure because they can actually introduce differences in how organisms can function. And sometimes 16S ribosomal RNA sequencing, therefore, is not enough. I presented an exciting new method to generate de novo genomes from metagenomes. And finally, I have shown you an example that suggests that intestinal microbes may be able to evolve in not only evolutionary timescales, but clinical timescales. So with that, I've tried to thank the people who were involved in the project along the process of giving you this talk, but I wanted to once again highlight the role of Eli Moss and Alex Bishara, who co-led this project, Alma Parada and Ann Dekis, who provided the ocean microbiome samples and were great collaborators, Serafim Batsoglu, who is a computational collaborator, and many others listed here. I'd also like to thank our patients. None of this work would be possible without their participation in our work, and of course, our funders. So with that, I'm happy to entertain some questions.
Okay, thank you so much, Dr. Bhatt. That was really wonderful. So we are opening up for questions now. Joining Dr. Bhatt, we have another panelist, Jill Hirschlook from 10x Genomics. So if you have a question for either Dr. Bhatt or for Jill, please go ahead and type it in to the Q&A panel. So you'll find the button here at the bottom of your screen. And if you push the button, it will open up a pop-up screen where you can type in your questions. And I will just give people a few minutes to start entering their questions. Okay, here's one, Dr. Bhatt. What is the minimum DNA input for this approach, both in terms of amount and molecular weight?
Great. So the minimum amount for input is 1 nanogram in terms of amount. In terms of molecular weight, the guidelines from 10x are having DNA input that's above 100 kilobases in molecular weight. For us, from a microbiome perspective, with standard extraction technologies, it's very difficult to achieve that level of DNA contiguity. So for our experiments, we've typically tried to target a 10 kilobase molecular weight cutoff. We find that having a 10 kb cutoff in terms of the blue Pippin prep provides us with contiguous enough DNA to get pretty good assemblies. Of course, it would be ideal to get higher molecular weight input, and we've been working on methods to do that in our lab. And we recently posted a preprint on bioRxiv. The lead author on that is Eli Moss, that reports a method to get much higher molecular weight DNA. We have yet to take that approach and apply it on the Linked-Read platform, but are actually actively doing that right now. So once again, to summarize, 1 nanogram of DNA input is enough, which is one of the great advantages of this compared to long-read sequencing approaches, which take usually on the order of at least 100 nanograms of high molecular weight DNA input. And we've been successful with size cutoffs, minimum size cutoffs of around 10 kb. But I would imagine it would do well even if you came down to 7 or 8 kb.
Okay, thank you. We have two sort of related questions here, so I'll combine them. So, and I'll have you answer this first, Dr. Bhatt, and then Jill might want to jump in as well. So how does Athena differ from supernova? Might it be helpful for human prokaryotic medium size, 1 to 40 kb structural variations that supernova failed to assemble? And then a related question is it possible to use this for bacteriophage metagenomics?
Yeah, so great questions. In our own efforts, we found that Athena far outperforms supernova in terms of prokaryotic applications, although technically with supernova 2, one should be able to get some assemblies. My guess is that for the applications that were suggested, especially the application of medium-sized structural variation that supernova fails to assemble, Athena would perform quite well. I will say in terms of the size cutoff, the larger the structural variation, you may actually end up needing differing minimum molecular weight cutoffs for your library preparation. But as you could see, we had a 60 kb structural variation that we were able to assemble into context quite well using a 10 kb cutoff library. And that really is because the big issue that we need to solve is the problem at the junctions. Fundamentally, the way Athena works is it essentially tries to pool barcodes and create smaller subassembly problems at the branches of the scaffold graph. And I think that's really the innovation in this particular approach. With respect to the second question, which was related to bacteriophage, I absolutely think this could be very useful in the context of bacteriophage assembly. I will say that some of the exciting and prevalent bacteriophages that are now coming to light, such as crassphage, which is a common human gut microbe or microbiome bacteriophage, and the megaphages such as laxphage, those seem to actually assemble really well using short read approaches alone. That said, there may be a lot of collapsing of variation in that process, and so I think it would be interesting to take linked read approaches to see if there are multiple strains or closely related strains of said phages in individuals.
Okay, thank you. Here's another question for you. Because often the microbes are uncultured or unculturable, how do you determine if a variant found during late stage of disease is truly a variant and not just a different strain population?
Yeah, I think this is a good question. To some extent, if I'm understanding the question correctly, the difference between a variant and a strain population is probably a semantic issue and a continuum in terms of what you call just a variant and what you call a strain, a different strain. It is difficult to really infer what the functions of these different strains might be without isolation and culture techniques. And one of the things that I think would be really interesting to do is to take an improved understanding of what these organisms can do and their metabolic sort of structure and function to try and improve methods to try and isolate and culture these organisms. So one of the advantages of actually having genome information for some of these unculturable microbes is that it might lend us some clues on how to actually isolate and culture them. And so those are at least some thoughts around that.
Okay, thank you. Let's see. So how does the DNA isolation change with different sample types?
One of the interesting things about different types of different domains of life is the cellular wall structure of these different domains of life really varies. So for example, if you were trying to study a microbiome sample or even just a pure microbial sample from a fungus, preparation of DNA from that sample would probably involve a chitinase or enzymatic approaches that will allow you to dissolve or degrade as much of the cell wall as possible. In the setting of mixed bacterial mixtures, we really usually approach this by using not only lysozyme, but lyticases that are derived from Aspergillus, et cetera. Once again, taking the first step to try and degrade the cell wall before we try and pop open the cell membranes. So I would say that the approaches to extract DNA from different samples really depend on what you think is in your sample. And one of the things that we found critical in this process is to use not only physical methods, but one way to minimize the use of aggressive physical methods is to use enzymatic methods that are well suited to the cell wall components that are present in the sample.
Okay, thank you. Next question here. Have you seen any other genome jumps besides the transposable elements in this dominant bug that could influence expression of genes?
So, we're in the process of actually trying to apply this more broadly to longitudinal samples from about 100 other patients from our collection. So I would say stay tuned for that. We have seen various examples of these transposable elements jumping in this particular organism. We've actually created some short read tools to be able to try and evaluate transposable element positions using isolate data. So as you guys probably know, there are these adaptive laboratory evolution experiments where people take E. coli, for example, and they expose them to antibiotics, and these organisms evolve antibiotic resistance. We've taken a look at data from those types of experiments and found that transposable elements can jump around the genomes of even organisms like E. coli and can confer antibiotic resistance by, for example, disrupting genes known to be important for antibiotic sensitivity, or upregulating genes that are important for antibiotic resistance. So we anticipate that this may be a generalizable phenomenon, and we actually have a preprint that focuses on that topic. That the lead author there is Matt Durant, that highlights what we think is maybe the broader biological role of transposable elements than has been previously understood.
That sounds great. I'll look out for that reprint for sure. The next question here, with 10x genomics, is it possible to quantify the number of genomes, for instance, for establishment of risk level of pathogens?
So I would say we're still in the process of actually getting this up to being a good workhorse technology in terms of being able to evaluate the number of genomes using the 10x approach. To be very fair, with our approaches so far, we can identify or generate genomes or near complete genomes from a large number of organisms from a sample, but there are some fixed challenges, which is that organisms that are poorly abundant are sometimes difficult to assemble. Organisms that have excessively repetitive elements in their genome, so organisms that have like 100 repetitive elements or a single element that's present 100 times can be really difficult to assemble. So I think we're a bit away from a comprehensive approach to generating, lots and lots, hundreds or even thousands of complete genomes using this approach. But I think there is an opportunity for additional technological development by our broader community to try and achieve that.
Okay, thank you. The next question here. What if more than one strain is responsible for any disease? How can an accurate interpretation be carried out?
That is a really good question. I think that at the end of the day, if more than one strain is responsible for a disease, meaning more than one strain is sufficient to induce disease, or more than one strain, meaning a combination of strains, is required for a disease, I really do think that genomics can't answer that question. In part because I think the genomic approaches are really good at figuring out what is present. We can do some advanced work to predict how these different types of genomes may be functioning, but at the level of trying to understand how strains are related to disease in a causal manner as opposed to an associated manner, I think it really requires orthogonal experiments, right? We need to do experiments that actually test causality and causal inferences. But it's a really important reminder for all of us that while these associations are very interesting, at the end of the day, we have to design other experiments that allow us to directly interrogate the phenotypes that these microorganisms might induce.
Okay, thank you. So here's another question about other potential methods. So would not long-read sequencers like PacBio, capable of reading up to 80,000 bases, eliminate all the problems related to the short-read platform?
So I think the long-read platforms, PacBio, Nanopore, are extremely powerful, and we have posted recent work that we've done applying the Nanopore platform. Of course, there are fixed problems with the long-read technologies. I think for us, the most significant issue has been, the first hurdle to come over has been getting enough DNA to be able to use those platforms. As many of you know, the long-read sequencing platforms require a higher amount of input DNA. And getting more DNA and getting high molecular weight DNA from some of these samples can be very difficult. These platforms also, at least in our hands, require higher quality DNA, so they're a little less robust than short-read sequencing technologies. The other major issue, of course, with these technologies is the error rate. So, of course, the error rate of PacBio is quite high, as is the error rate of Oxford Nanopore technologies. The PacBio errors are essentially kind of random, and so you can do a lot of additional sequencing to essentially smooth over those and get a higher accuracy consensus sequence. In the setting of Nanopore, the errors are not random, right? They have an issue with homopolymer repeats. So at least in our hands, when we've done nanopore-based experiments, we've required short reads in order to polish up the accuracy. So we need an orthogonal method. One of the nice things about linked reads, in addition to having low input, meaning if you don't have a lot of DNA or you're not able to get a lot of high molecular weight DNA, you can still use it. The other advantage is, of course, the high short read accuracy. So you don't need to do an orthogonal short read sequencing experiment. You can use your 10x data to do the seed assembly and all subsequent steps, but also you don't have the error rate that you do in these other methods. So I'd say those are the circumstances in which the linked read approach is superior. Of course, if you just know that, for example, genes are linked together in 10 or 100 kb, and you don't have to do fancy bioinformatics or try to infer that, that is advantageous. But I would say that there are use cases for each of these technologies, depending on what you have as input and what you're trying to get out.
Okay, thank you. And we have a question here that I'm going to toss your way, Jill. How many fragments would the majority of drops contain?
I'd have to look up the precise answer, so I can email that to you later. But I believe for a standard reaction, if you add 1 nanogram of input to the reaction, each droplet will have around 150,000 base pairs worth of DNA inside. So if your fragments were 50 kb, you'd have a few molecules. And that's how we design our system. It doesn't cause any adverse effects. I think a lot of people think there's 1 molecule per droplet and that's necessary to get linked read behavior. It's actually not the case. We have handfuls of droplets present per molecule.
Okay, thank you, Jill. Here's another interesting question, Dr. Bhatt. Is it possible to get the sequence of plasmids associated with bacterial genomes?
So that is actually one of the blind spots of this type of technology. As it stands, plasmids, of course, can be assembled and assembled separately, and oftentimes we get really nice circularizable contigs. Unfortunately, trying to map those back to the chromosomal DNA is difficult. I think there are orthogonal approaches that could be really valuable. One is a single-cell sequencing approach, and while we haven't done this yet, we're really excited to try and leverage single-cell sequencing approaches to try and map plasmids to genomes, chromosomes. Of course, high-C approaches can be useful in this regard as well, and it has been demonstrated, at least in terms of proof of concept, that plasmids can be mapped to chromosomal DNA using high-C approaches. Of course, the high-C approach, at least in our hands, has required a lot of additional sequencing, and the data are complex to deconvolve. And so that is one of the challenges of that approach. Other approaches that have been proposed and I think are really interesting is looking at things like methylation marks on the plasmid DNA and looking to see if you can match methylation marks on the plasmid to the host chromosome. Of course, that would require, either something like bisulfite sequencing or probably even more advanced types of modification or more advanced methods to look at N6-adenine methylations, which are the most common methylation in bacteria, and obviously bisulfite sequencing does not capture that, or direct modification measurement approaches that is technically possible using the PacBio platforms and the Nanopore platforms. So I think that's a really important thing for all of us to be thinking about. Those are some examples of how we might approach that are isolation and culture independent, but I think there's opportunity to develop all of those.
Okay, thank you. I just want to give you one update. Jill just informed me that she checked our documentation, and it's 450,000 base pairs per droplet. You guys want to drop that one down. We're not going to be able to get to all of the questions because we've reached our stopping time, but I just want to remind everybody that about a week or two, we'll have a on-demand version of this recorded and on our website, and you're welcome to contact the 10X support team with any other questions that you might have. Thank you so much, Dr. Bhatt, and have a wonderful day, everyone.